USMLE, Step 1. Year 2024, number 0958. A 34-year-old woman comes to the physician because of a 2-day history of mild nausea, increased urinary urgency and frequency, and constipation. She also has had a 4.5 kilograms 10 lb weight loss during the past 2 weeks and a 3-week history of vaginal bleeding. Pelvic examination shows a nodular cervix with an irregular, friable posterior lip, and a rock-hard, irregular, immobile pelvic mass that extends across the pelvis. Examination of biopsy specimens from the cervix and anterior wall of the vagina show well-differentiated keratinizing squamous cell carcinoma. Which of the following best describes the pathogenesis of this patient's disease? A. Transactivation of cellular growth factor genes by tax. B. Specialized transduction. C. Insertion of viral promoters adjacent to cellular growth factor genes. D. Inactivation of cellular p53. E. Translocation of CMYC to an Ig gene promoter. Types of genes playing a role in cancers. A cancer is often the result of changes in more than one of these types of genes within a cell. Tumor suppressor genes, oncogenes, DNA repair genes. A tumor suppressor gene, anti-oncogene, is a gene that regulates a cell during cell division and replication. When it is mutated, it results in a loss or reduction in its function. And in combination with other genetic mutations, it can cause the cell to grow uncontrollably, resulting in cancer. An oncogene is a gene that has the potential to cause cancer. In tumor cells, these genes are often mutated, or expressed at high levels. Activated oncogenes can cause damaged cells to survive and proliferate, instead of apoptosis. Examples of tumor suppressor genes. Functions. Associated carcinomas. P53, tumor protein P53. Apoptosis. Half of all known malignancies. RB, retinoblastoma protein. DNA replication, cell division, and death. Retinoblastoma. VHL. Von Hippel-Lindau. Cell division, death, and differentiation. Kidney cancer. APC. Adenomatous polyposis coli. DNA damage, cell division. Migration, adhesion, death. Colorectal cancer. BRCA2, breast cancer 2, cell division and death. And repair of double-stranded DNA breaks. Breast cancer, ovarian cancer. NF1, neurofibromatosis. Type 1, cell differentiation, division, development, raw signal transduction. Nerve tumors. Neuroblastoma. PTCH, patched, hedgehog signaling. Medulloblastoma, nevoid basal cell carcinoma. Syndrome. NBCCS, Gorlin syndrome. P53 is a tumor suppressor. It can prevent cancer formation by conserving stability by preventing genome mutation. TP53 gene encodes proteins that bind to DNA and regulate gene expression to prevent mutations of the genome. The TP53 gene is the most frequently mutated gene, greater than 50%, in cancer. RB is a tumor suppressor protein. One function of RB is to prevent excessive cell growth by inhibiting cell cycle progression until a cell is ready to divide. When the cell is ready to divide, RB is phosphorylated, inactivating it, and the cell cycle is allowed to progress. It is also a recruiter of several chromatin remodeling enzymes such as methylases and acetylases. It is dysfunctional in several major cancers. Human papillomavirus, HPV, is the most prevalent sexually transmitted infection in the world. Few people even know they are infected, as the symptoms are seldom noticeable. Nearly all cervical cancers are directly linked to previous infection with one or more of the oncogenic types of HPV, e.g. types 16 and 18. HPVs have circular, double-stranded DNA genomes that are approximately 8 kilobits in size and encode 8 genes, e.g. E6, E7. The viral E6 and E7 oncoproteins have transforming properties and pleiotropic functions, such as transmembrane signaling, regulation of the cell cycle, transformation of established cell lines, immortalization of primary cell line, and regulation of chromosomal stability. They are necessary for malignant conversion. Transformation is the genetic alteration of a cell resulting from the direct uptake and incorporation of exogenous genetic material from its surroundings through the cell membranes. The recipient bacterium must be in a state of competence, which might occur in nature as a time-limited response to environmental conditions such as starvation and cell density, and may also be induced in a laboratory. It leads to horizontal gene transfer. Types of horizontal gene transfer: conjugation, transfer of genetic material between two bacterial cells in direct contact; transformation, exogenous genetic material passes from one bacterium to another; transduction, injection of foreign DNA by a bacteriophage virus into the host bacterium; vesiduction, transfer of bacterial DNA by extracellular vesicles. E6 consists of 158 amino acid residues and contains 2 zinc finger binding motifs and promotes cell proliferation by stimulating degradation of the tumor suppressor p53 protein via the formation of a trimeric complex comprising E6, p53 in the cellular ubiquitination enzyme E6AP, leading finally to increased tumor cell growth. RB is normally functions as a tumor suppressor to prevent excessive cell growth by inhibiting the activity of transcription factor E2F. By binding to RB, E7 induces a conformational change in RB, leading to the release of E2F transcription factors that RB normally keeps inactive. This activates genes necessary for DNA synthesis and cell cycle progression. The cell is prompted to enter the S phase of the cell cycle, leading to DNA replication and cell division and inhibition of apoptosis. Apart from disrupting the cell cycle control, E7 can also interfere with pathways that lead to apoptosis, further promoting cell survival and proliferation. These processes can lead to genomic instability and result in carcinogenesis. The answer is D. Inactivation of cellular p53.